The core of the scientific method relies on a single, uncompromising rule. If a discovery is real, independent researchers must be able to follow the exact same steps and get the exact same result. Scientific truth is geographically independent. A biological mechanism discovered in an Auckland laboratory has to perform identically when tested by a team in Berlin. Without this repeatable proof, an experiment is indistinguishable from an anecdote. A single study, no matter how impressive the findings, proves nothing on its own. This repetition serves as the quality control mechanism for the modern world. It is the filter that validates the medicine we take and the technology we rely on. In the early 2010s, biotechnology giants Bayer and Amgen attempted recreating dozens of landmark preclinical cancer studies to provide a base for new drug development. The results were staggering. Independent teams attempting to repeat experiments confirmed only 11 to 25% of high-impact oncology findings. By 2015, the Reproducibility Project successfully recreated just 36 of 100 prominent psychology studies. This collapse spanned across multiple disciplines. The replication crisis means we're designing medical treatments and social systems on foundations that have never been verified, and in many cases are likely false. Modern science functions as an industrial economy, where a researcher's funding, tenure, and very livelihood depend on their volume of publication. Prestigious journals act as the gatekeepers of this economy, but they rarely publish standard confirmations. They prioritize novel breakthroughs and exciting positive results. This creates a problem for null results, studies that prove a drug or a theory simply doesn't work. Because journals find these failures boring, researchers often shove them in a drawer. This publication bias leaves the historical record filled with statistical flukes, while the evidence of what doesn't work disappears. The institutional system accidentally rewards a headline-grabbing story over a verifiable, repeatable truth. To survive in this economy, researchers often fall into the trap of p-hacking, an unconscious survival tactic used to turn a failed study into a publishable one. Here's how it works. Researchers start with chaotic data, then begin dropping outliers or shifting parameters. Eventually, the random noise is manipulated until a statistically significant trend line emerges. Sometimes a researcher will even retroactively change their original hypothesis to match whatever result the manipulated data finally coughs up. These are rarely acts of intentional fraud. They are systemic blind spots, small, culturally accepted tweaks that allow honest scientists to produce the results the system demands. The intense pressure to be right leads researchers to unintentionally torture their data until it confesses. Even when someone tries to verify a study, they often find the original blueprints are missing. In 2021, a project attempted to replicate high-impact cancer biology experiments, but struggled to even find the instructions. Across the studies they analyzed, exactly 0% of the original protocols were fully described in the published papers. They also hit a human bottleneck. 32% of the original authors were unresponsive or unhelpful when asked to share their reagents or specific cell lines. A replication attempt is doomed to fail if the original architects refuse to share their methods. This crisis gave birth to Metascience, the practice of turning the scientific method back on science itself to diagnose and repair these institutional cracks. The first major fix is the rise of registered reports and pre-registration. This process requires researchers to publicly archive their hypothesis and methodology before they ever collect data, making it impossible to p-hack the results later. Researchers at Northwestern University are now using machine learning to catch biases that humans miss. This AI scans the text of thousands of papers, looking for linguistic cues that predict if a study can actually be replicated before anyone spends the resources to retest it. These tools are shifting the institutional focus away from flashy anomalies and toward radical transparency. While the 75% failure rate in oncology is a disaster for individual theories, the fact that science identified its own rot is the system's self-correction mechanism in action. Science earns its credibility because it is the only institution that systematically refuses to trust itself. This transition toward open, networked science makes it significantly harder to hide data in a drawer or manipulate a result for a headline. The replication crisis provided the evidence needed to overhaul academic incentives, forcing a shift toward more transparent and verifiable research.